Lyme disease is caused by a bacterium called Borrelia burgdorferi. Our lab is focused on trying to eradicate this disease. One of the things we're interested in is how the bacteria uses ticks to move around between different animal hosts. The bacterium is an obligate parasite, which lives off the nutrients provided to it by the animals it lives in. These hosts are small vertebrates such as mice, squirrels, and birds. The bacteria cause infection as they move around looking for new hosts to feed off. Ticks are considered a vector as they help to transmit the bacteria to other vertebrates. In a more simplistic explanation, bacteria are essentially hitching a ride on the tick to find new hosts to infect. An unintended consequence of the bacterium hitching a ride on the tick is that they can encounter humans. This interaction can spread the bacteria to us and can ultimately lead to disease. However, on a more positive note, the ticks aren't always infected themselves. An adult female cannot pass the bacteria to her offspring. This biological block is a critical step in helping us to eradicate Lyme disease. This means that if we can stop the movement of Borrelia from chronically infected animals into ticks, then we can eventually stop Lyme disease. But how do we do this? The bacteria must adapt to their different environments in order to survive. We are working on how the bacteria protect themselves as they move between the ticks and the animals to be more successful at moving from host to host. Here I will detail a few of the differences between the major hosts, the tick and the mouse. The first being that the tick is an arthropod while the mouse is a mammal, containing different immune systems. Second, the tick only feeds a few times throughout its life cycle, which means the bacteria must be able to survive prolonged periods of starvations. While within the mouse, they have access to a more consistent source of food. Another major difference is the temperature that the bacteria are exposed to in each host. The tick can be outside through the winter and experience freezing temperatures, while within the mouse, the bacteria is exposed to relatively consistent high internal temperatures. We think that if we can find a critical step in the adaptation to the tick, then we can block it. If we can block it, then the bacteria won't be able to reinfect new ticks each year. If the bacteria are unable to get from the vertebrates into the tick, then they will not be able to transition from one host to another. Once we find a drug that can prevent transmission to ticks, then we can start giving it to mice.
I just want to give you an idea of how we are trying to search for good drug target candidates. We're utilizing high throughput sequencing along with tagged strains of the bacteria to identify vulnerabilities in the bacteria at a higher efficiency than before. This will hopefully lead to a faster drug discovery process. I really hope I don't have to run around the Northeast trying to spoon feed the medicine to mice, although that's a sacrifice I'm willing to make. Ultimately, if we decrease the number of infected ticks, then we will decrease the number of interactions between humans and ticks that contain the bacteria. Over time, this will lead to a decrease in Lyme disease and hopefully a complete eradication of this disease in the future.